So how does the cell regulate which proteins it makes? Now, there are some proteins that all cells need to make, and these are the so-called housekeeping proteins, and they are encoded on housekeeping genes. These proteins are typically used in processes that are common in all cells, processes such as protein synthesis and energy production. However, the cell doesn't need to express all the proteins that are in their genomes. This type of regulation can be referred to as spatial regulation, in that different cell types, which will be in different parts of the body or organ, will make the different proteins they need for their function. The other type of regulation is temporal or time regulation. That is, a cell only needs to make certain proteins at certain times. These times could be due to development, that is, as the organism develops, differentiation, that is, as the cells specialize. They could be cell cycle dependent, as different proteins are needed at different stages of the cell cycle. Or they may be inducible, that is, the body only triggers their production when they are needed. Okay, so now we're going to look at gene regulation and protein expression. Well, there are 6 key points in the process of making a mature, fully working protein that a cell can control. And they are in order of making the protein. 1. Transcriptional control. Does the cell make the RNA or not? 2. RNA processing. How is the RNA processed to make the final messenger RNA? 3. Transport and localization. Do we transport the messenger RNA from the nucleus, and where does the messenger RNA go in the cell? 4. Translational control. How is the protein translated? Where in the cell and how many times? 5. RNA degradation. How long is the messenger RNA available for translation? How quickly is it degraded? And 6. Protein regulation. Is the protein activated and or modified in some way? Just upstream from the gene is the core promoter region that contains the tartar box. And upstream of the core promoter region is the regulatory promoter region. And it is this regulatory region that the cell uses to control transcription of the gene. Using the regulatory region, the cell has 4 main ways of controlling transcription. It can use tissue-specific transcription factors that bind to the promoters in the regulatory region. Or it can use inducible transcription factors that either bind to the DNA sequence directly or act via cell signaling pathways to affect transcription. Or it can use alternative promoters, that is, 2 different promoters for the same gene. And then finally, the cell can exert control through epigenetics, which is the chemical modification of the DNA to control transcription. So, looking at the first method, specific transcription factors. The cell has a number of common transcription factors that can be found in all cells. However, the cells can also have tissue-specific transcription factors. This way, the cell gains control through the production of the correct transcription factor as to which genes are transcribed. Next, the second method, the method of transcription control through inducible transcription. In this control mechanism, the genes are only induced as a result of an external stimulus, such as hormones, heat shock, or nutrients. These stimuli can activate their specific transcription factors and start the transcription of the genes required to respond to that stimulus. Number 3 is alternative promoters. And as the name suggests, this is the use of alternative promoters within a gene. With alternative promoters, you can get the same protein being produced, but the transcription triggered as a result of different circumstances or conditions faced by the cell. With the use of alternative promoters, the cell can make the usual protein, or one with a different N-terminus, or a completely different protein. Finally, epigenetic control, which is the chemical modification of DNA so that transcription is regulated. Interestingly, this is a modification that can be passed on from one generation to the next, a process known as genomic imprinting. In this method of control, methyl groups are added to the DNA, and these can inhibit the gene promoter. Methyl and acetyl groups can also be added to the histones. In this case, the methyl groups inhibit or silence transcription, and the acetyl groups can activate transcription. In terms of RNA processing, the cell can exert control in three different ways. Alternative splicing, for example exon skipping, alternative polyadenylation, which is using a different site for the poly A tail, and 3, messenger RNA editing, which is doing a different RNA edit in different tissues to give tissue-specific proteins. In alternative splicing, there are 5 methods that the cell can use. 1, exon skipping. 2, intron retention. That is, the cell keeps the intron in the messenger RNA, which then adds additional amino acids to the protein. 3, alternative 5' splice site. Essentially, the cell keeps the 5' end of an intron and excludes the 3' end. 4, alternative 3' splice site. And this is just like the 5' splice site, but instead the cell drops the 5' end of the intron and keeps the 3' end. And finally, 5, mutual exon exclusion, which is when 2 exons can't both be present in the final protein. That is, it is either one or the other, but not both. In alternative polyadenylation, the pre-messenger RNA will contain 2 sites for polyadenylation, one weak site and one strong site. And to meet the current requirements of the cell will pick which one it uses. Alternative polyadenylation causes changes to the C-terminal of the protein and can also change how long the mature messenger RNA is available for translation. That is, it can impact the degradation of the messenger RNA by the cell. Finally, control 3, tissue-specific editing. Here, as the name suggests, the cell exerts control depending upon the tissue that the cell is in. Typically, the RNA will fold by pairing with itself, usually in an intron, and an A will be deaminated to give inosine. And this will mark the site for removal. Sometimes in this process, a C will be converted to a U to give a stop codon. Either way, the result is an edited messenger RNA. After modifying the pre-messenger RNA to give the required mature messenger RNA, the next level of control the cell has on the production of the protein is where the messenger RNA ends up in the cell. The messenger RNA, which is produced and matured in the nucleus, can either randomly diffuse through the cytosol or can be transported to a specific location. And it is the 3' untranslated region that plays a role in this process. As you will recall, the 3' UTR is the region between the stop codon and the poly A tail, and this region contains a number of RNA sequences that impact the export, localization, half-life, the amount of time the RNA is available for translation, and the production of the protein. The peptide that first emerges from the ribosome messenger RNA complex can also dictate where the protein is made in the cell. The next stage in the journey of the production of a protein from a messenger RNA is to make the protein, translation. Now, 2 terms we need to address are cis-acting and transacting. In biomedical sciences, when we say cis-acting, it just means within the same molecule, whereas transacting means between different molecules. In terms of what we're talking about, an example of a transacting element could be a protein that is produced by one gene and which either activates or inhibits the production of a protein by another. An example of a cis-acting element are sequences in a DNA or RNA molecule that interact with transacting elements to control transcription or translation. The sequences are in the same molecule, so they are cis. The untranslated regions of messenger RNA contain cis-acting regulatory sequences, and these sequences can be targeted in 4 different ways. 1, transacting RNA binding proteins to promote or regulate the longevity of messenger RNA or block the production of the protein. 2, by external stimuli. 3, by small molecule binding to the messenger RNA, which can cause a refolding. And 4, by microRNAs, which can bind and then either inhibit translation or promote the degradation of the messenger RNA. Upstream of the start codon in the 5' UTR, the RNA molecule may be able to fold back on itself through the pairing of its bases. This folding or formation of loops and hairpin loops can give specific binding points for the proteins to interact. Also, small molecules and ions can also act at these points in the messenger RNA. An example of this is when a messenger RNA folds to give a binding site for a protein that can then block translation. This is an example of a transacting RNA binding protein. Another example of how these folds can regulate translation is through the action of an external stimulus such as heat. In this case, the RNA loop in the 5' UTR is blocking translation, and the temperature increase causes the loop to melt, which just means causes the loop to unfold. This melting then allows the translation machinery access to the start codon. This is a type of regulation that you might find in plants. For small molecule binding, the presence of a small molecule could cause the RNA to fold in a slightly different way in the 5' UTR. This change in folding could then block the start codon and therefore block the translation. Once the small molecule is no longer available, the RNA then refolds, revealing the start codon, and hence the protein can be made. Finally, for microRNAs, the microRNA could just simply bind to the messenger RNA and block the start site. Well, another way that a cell can control protein production is by the length of time the messenger RNA is available to make the protein. A messenger RNA in a human cell typically has a half-life, the length of time it takes for half of it to be degraded, of around 8 to 10 hours. However, some messenger RNAs may only last 30 minutes and some may last more than 10 hours. And the cell can control how long messenger RNA is around before it is degraded. The eukaryotic cell has two mechanisms to destroy messenger RNAs. It can either remove the 5' cap or degrade the molecule from the 3' end. Both processes start with the shortening of the poly-A tail by an exonuclease. You can view the poly-A tail as a timer because the exonuclease will shorten the tail over time. And when the tail gets to around 25 A's, the messenger RNA will be uncapped, causing a rapid degradation of the 5' end, and the degradation will also be continuing from the 3' end. The system that controls the half-life of the messenger RNA are things like chemical modifiers that mark the RNA for destruction, and sequences in the 3' untranslated region which act as binding sites for proteins or metal ions that can increase or decrease the half-life of the molecule. When the pre-messenger RNA is being produced by the cell, transcription factors and DNA promoters can influence the stability of the final messenger RNA by chemically modifying the molecule as it is produced, and either marking it for early breakdown or for increased longevity. An example of increasing the longevity of the molecule is by putting more A's in the poly-A tail, which would mean the exonuclease would take longer to get to the 25 A's, the point at which degradation and uncapping starts. The sequences in the 3' UTR that could control the longevity of the molecule may form loops that play a role in controlling the stability of the molecule by interacting with proteins or other molecules or even ions, or the sequences may act as binding sites for microRNAs that can influence the degradation of the molecule. The cell can control gene expression at the protein level in a number of ways. First, the cell exerts control by determining where the protein is produced. That is it produced in the cytosol or the endoplasmic reticulum? The default pathway is in the cytosol, and it is a sequence in the end terminal of the protein that says the protein should be produced at the endoplasmic reticulum. The cell can also control whether the protein is active, that is, whether or not it needs some modification to become fully functional. These modifications are often referred to as post-translational modifications, although interestingly, some of them can occur while the protein is still being made. Examples of such modifications are cleavage, the removal of a peptide to make the protein active or inactive, the addition of inhibitor proteins, glycosylations, the adding of sugar groups to protect the protein from degradation or to help target it to the correct part of the cell, phosphorylations, the addition of phosphate groups, which can have a range of outcomes such as activation, deactivation, controlling interactions with other proteins, and even targeting the protein to a different part of the cell. And finally, the ultimate level of control, marking the protein for degradation through the addition of certain molecules such as ubiquitin. We have covered a lot of very important stuff quite quickly. We've looked at the 6 levels or stages of protein production where control can occur. That is, we've looked at transcriptional control, RNA processing, transport and localization, translational control, RNA degradation and protein regulation.